Okay, now you can start.
Okay. So my presentation is about cryogenic electron microscopy. This is one of the methods that scientists use for structural investigation. This is my outliner. First, I talk about importance of the topic and some techniques for structural biology. And then I explain two cryo-EM method, which is single particle cryo-EM and cryo-electron tomography. So this slide is about the importance of topic. As you can see, according to World Health Organization, at 2050, 15% of total population of the world will be old or older population. And growing old has its own problem like chronic disease such as Alzheimer, Parkinson, diabetes, and so on. So preventing and managing this disease is a global concern. Structural biology is one of the techniques that study chronic disease. To answer how, we need to talk about biology. This is overview of biological hierarchy, and in structural biology, we work to link the connection between the 3D structure and molecular function. In this technique, we work to link what it looks like to help understand how it does it. Imagine you have a fork and a spoon. If you can see this shape and structure, somehow you can guess what is their function and how you can use them. So macromolecule in our body is same. They need to be in a specific shape and structure to be functional. If their shape change, their structure, their function change to and leading to chronic disease. So first step to study disease is study the structure of the molecule. This is some structural biology techniques. As you can see, X-ray crystallography, cryo-EM, and nuclear magnetic resonance is a most popular technique for structural biology. And in the right side, you can see a chart, and you can choose which method is the best method for your molecule according to structure size. So let's talk about cryo-EM. This is 3 Nobel Prize winner in chemistry in 2070 for their contribution to development of cryo-EM. And with a recent progress in detector technology and software program algorithm, now we can have a structure at near atomic resolution around 3 angstrom, which is amazing. And in the right side, you can see the total number of structure found by cryo-EM in Protein Data Bank PDB database, which had reached to 3,000 in 2019, and this growth rate is expected to accelerate in future. So first question is, why cryo-EM? First things is, in cryo-EM, we don't need to crystallize our protein or macromolecule, like X-ray crystallography. Crystallization process is very time consuming, and for some protein, like big protein, it's impossible to do this. The second of all is it has a low radiation damage. As we use this method in the low temperature, ice acts as a protective layer for our sample and somehow lower the radiation damage into our sample. And also we can capture our molecule structure in the native state, which is very important when we want to study the function of it. This method also requires a small amount of the sample, unlike NMR. Sometimes our sample is very expensive, or sometimes it's hard to get. And another one is you can use this technique for complex structure and large molecule weight. You can use for 150 to 300 kilodalton molecule, and also with the recent progress of this field, it's expected even to be a replacement for X-ray crystallography, and for now, in one field, even in this technique is better than other techniques, which is for membrane protein. And in this chart, you can see success rates vs protein structure complexity. So let's talk about different type of cryo-EM. First one is single particle cryo-EM. As you can imply from the name, we study single molecule in isolation here. And the second is cryo-electron tomography, which we study macromolecule in situ, in its own environment. So first method, this movie shows the step for SPA. Okay, sound easy, right? But no, there is many obstacles and challenges you may encounter when you work with cryo-EM. Challenges like thin ice layer orientation bias, particle partitioning, structure heterogeneity, beam-induced motion, electron radiation damage, and so on. So I try to cover some of these challenges and give some solution for it. The first one is electron radiation damage. This is the overview of the electron microscopy design. As you can see, it has some coil, like electromagnetic coil that redirects our electron to the right path to our sample. And this device works with wave particle duality of electron, and this is the fundamental formula in quantum mechanics. And with a typical voltage of 300 kilovolts, we have a wavelength around 0.02 angstrom. If we compare it with size of atom, which is in order of angstrom, we can see many details in our cell. But there is one problem, which is biological samples are very sensitive to electron radiation. As I said before, in this method, we use ice as a protective layer to protect our sample from electron radiation damage. But it's just a matter of time. After a while, our sample will be cooked by electron radiation. So one solution for this is using a low doses electron beam. Another one is structural heterogeneity. In order to make a good 3D model, we need a good distribution of our particles. That means we need particles with same size, mass, and shape. But what if instead of the left picture, we have the right picture. Here, it's almost impossible to average them and make a good model from it. So what we need to do before doing any cryo-EM process and experiment, we need to purify our sample with some chemical experiment. Another thing is some of the protein has flexible domain. That means their movement also makes some heterogeneity in our image. So what the scientists do using small molecules, inhibitor, cofactor, or anything can stabilize our protein and make it a good particle for imaging process. The third challenge is orientation bias. So as you see from the steps, we need to average our particle image to make a 3D structure. So we need different view and orientation to make that. But what if instead of the left picture, we have right picture? Here we have our particles are adapted to very limited orientations. So to solve this challenge, there is two solutions. One of them is using detergent. The cause of having orientation bias mostly is because our particle interact with air-water interface. So when we use our detergent, somehow we make a layer, and as this layer occupied the surface of this air-water interface, our particle don't have any space to go there and interact here. Another one is tilting our sample. So this is more easier than the first one. So imagine you have a limited orientation of the particles, but what you can do is take a first picture and tilt your sample, and take a second picture and tilt it, and so on. So with the repeated process, although our particles are adapted to very limited orientation and view, we have a 360 view of our particles. And the last thing, in order to evaluate our particle and their distribution and purity, we need to do negative stain before doing any cryo-EM. So this movie somehow explain what is this step and what is a negative stain experiment.
Virtually every cryo-EM project is preceded by evaluating the particles of interest using negative stain TEM. This relatively simple and quick method enables users to gauge particle quality and provides a first-order estimation of feasibility of their cryo-EM project. First, we need to prepare the grid by glow discharging. The surface of the grid is carbon-coated and tends to be hydrophobic. Glow discharging will make the surface of the grid hydrophilic, allowing our sample to spread easily. Next, 2 to 4 microliters of sample is pipetted onto the prepared grid and allowed to adsorb to the surface. After around a minute, the excess sample is blotted away using filter paper. The grid is washed with a droplet of water, dried again, and then the stain is applied. The stain, in this case 1% uranyl acetate, is allowed to adsorb for about 20 seconds. The excess stain is blotted away and the grid is left to air dry. If we were to view what is happening on the grid during the sample preparation stage at the nanoscale level, we would see the sample getting deposited onto the carbon film with a protein landing in random orientations. Now the excess sample is removed by blotting. The stain is then added. The stain particles uniformly coat all surfaces, covering the proteins and the carbon film. The excess stain is then removed and the sample allowed to air dry. Once the sample is dried, it's ready to be viewed using the TEM.
Okay, so although the workflow is very high, it does worth the time and effort. This is two examples of single particle analysis. As you can see, after just 3 weeks, this two group that received the genome of the SARS-CoV-2, they had the structure. So let's talk about the second method, which is cryo-electron tomography. In this method, also we are interested in what's happening in the cell, but in its own environment. So here, because cell is a very gigantic object, we cannot just use low temperature. We have to use three things all together, which is low temperature around -150 centigrade, high pressure, which is around 2000 bar, and very fast. So as you can see from this chart, we are in amorph ice region, and these amorph things help us to see the structure of our macromolecule vividly and clearly. And the last picture shows the freeze-fresh liver cell. This last movie also showed the step for cryo-electron tomography. And this is the example of cryo-electron tomography. This type of algae. As you can see, we have a very beautiful structure in its own environment. Okay, in summary, structural biology is a method for studying chronic disease, and these techniques help us to understand the link between 3D structure and molecular function. Cryogenic electron microscopy is one of the structural biology techniques which can provide 3D model of our macromolecules. And certain methods have been developed to overcome the different challenges that arise in cryogenic electron microscopy.
Great, thanks very much, Fatma. Really interesting, nice visuals. That was very, very cool.